Hello and welcome to Medical Dialogues. I'm Anshika Mishra. Here's what I bring to you from the world of medicine. Research discovers microplastic in every human placenta. In a study published on February 17, 2024 in the journal Toxicological Sciences, a team led by Matthew Campen, PhD Regents Professor in the UNM Department of Pharmaceutical Sciences, reported finding microplastic in all 62 of the placenta samples tested, with concentrations ranging from 6.5 to 790 micrograms per gram of tissue. Microplastics are present in virtually everything we consume, from bottled water to meat and plant-based food. Additionally, research has shown that these particles have infiltrated the human body, with microplastics detected in organs and tissues. While microplastics are already present in our bodies, it is unclear what health effects they might have. Traditionally, plastics have been assumed to be biologically inert, but some microplastics are small, measured in nanometers, and are capable of crossing cell membranes. The growing concentration of microplastic in human tissue might explain puzzling increases in some types of health problems such as inflammatory bowel disease and colon cancer in people under 50, as well as declining sperm counts, said Campen.
Are you also aspiring to excel in medical journalism and delve into the intricacies of health and medical reporting? Explore our comprehensive course on medical and health journalism available at the Medical Dialogues Academy. Learn from industry experts who possesses in-depth knowledge and experience in this field. For further details, scan the QR code provided or access the link in the description box below.
Red light has the potential to decrease blood glucose levels, studies suggest. A study published in the Journal of Biophotonics indicated that red light has been shown to decrease blood glucose level by 27.7% after glucose intake, as well as reduce maximum glucose spiking by 7.5%. Mitochondria, the cellular powerhouse, utilizes oxygen and glucose to generate the energy-rich nucleoside adenosine triphosphate, which is the ATP, vital for cellular processes. Previous research has demonstrated that long wavelength light ranging from approximately 650 to 900 nanometers, spanning the visible to near infrared spectrum, can enhance mitochondrial ATP production. This in turn decreases blood glucose level and promotes improved health and longevity in animals. Additionally, researchers discovered that red light at 670 nanometers stimulated energy production within mitochondria, resulting in heightened glucose consumption. To study the effects of 670 nanometer red light on blood glucose levels, the researchers enlisted 30 healthy participants. These participants were randomly assigned to two groups, 15 in the 670 nanometer red light group and the 15 in the placebo, that is no light group. Excluding individuals with known metabolic conditions and those taking medications, the participants underwent an oral glucose tolerance test. They were then instructed to monitor their blood glucose level every 15 minutes for the following 2 hours. The findings revealed that people who received red light exposure 45 minutes before drinking glucose exhibited a reduced peak blood glucose level and reduced total blood glucose during the 2 hours. Oocytes cleverly evade toxic proteins to maintain long-term female fertility. Researchers at the Center of Genomic Regulation in Barcelona have discovered a new mechanism which explains how oocytes remain in pristine condition for decades without succumbing to the wear and tear that would cause other cell types to fail. The research published in the journal Cell represents a new frontier to explore the unexplained causes of infertility. Oocytes are immature egg cells that develop in almost all female mammals before birth. The propagation of future generation depends on this finite reserve of cells surviving for many years without incurring damage and can last almost half a century, the average time between birth and menopause. The researchers studied protein aggregates, which are clusters of misfolded or damaged proteins known for their toxic effect. These aggregates accumulate in the cytoplasm and are associated with various neurodegenerative diseases. Cells typically manage them by enzymatically breaking them down or segregating them during cell division. However, oocytes, responsible for contributing their entire cytoplasm to an embryo after fertilization, reduce their metabolic activity to minimize the generation of harmful byproduct that could compromise maternal DNA and future reproductive successes. As a result, oocytes are particularly sensitive to misfolded or damaged proteins. Thank you for watching. Stay tuned to Medical Dialogues for more updates.
Never miss a medical update from Medical Dialogues. Like, subscribe, and press the bell icon.